get started promptly. Thank you all for coming. I'm going to present on the genomics of adaptation in an emerging marine annelid model, which is Striplospio benedicti. So, despite myriad differences in evolutionary history and ecology in marine invertebrate larvae, two life history modes predominate. Namely, this is planktotrophy on the left and leucithotrophy on the right, and they represent co-evolved suites of organismal traits, and many represent adaptations to the niche of the larvae. These distinct larval syndromes are complex, and their evolution involves changes in cellular, developmental, and behavioral phenotypes. So, for instance, the transition between small and big eggs involves differences in maternal provisioning, and then the difference in feeding from plankton to yolk involves metabolic differences. This makes it surprising that these shifts in larval life history, usually from planktotrophy to leucithotrophy, occur frequently within and between animal taxa and in relatively short periods of time. And these two life histories likely represent peaks on the adaptive landscape, so one would imagine that these organisms would have to go through the fitness valley in order to go to either side. So how these transitions happen remains unclear. My model organism, Striplospio benedicti, has the largest heritable intraspecific variation in oocyte size among animals that we know of. As you can see in this picture here, you have planktotrophs on the top, which produce small eggs, and leucithotrophs on the bottom. And different genotypes of females can produce oocytes that vary up to 8-fold in volume. Just to break this down a little further for you, planktotrophs emerge from numerous small eggs and feed in the water column until metamorphosis, and their larvae exhibit complex anatomy, which again are likely adaptations to their period of growth in the pelagic zone, where they have to deal with predators and locomote in the water. On the other hand, leucithotrophs emerge from few well-provisioned eggs and settle shortly thereafter into the benthos, and they have comparatively simple anatomy. When you look at the phenotypic variation of the larvae, this is, I phenotyped larvae from over 400 females that I collected from the wild, and I essentially plotted a characteristic of the larvae on the y-axis, which is basically how much bristles they produce, and on the x-axis is larval size. And you can see that the larvae demonstrate a strong dimorphism, even though, as you can see when I color it by geography, different morphs co-localize in the same locality. So our organism provides us with the opportunity to find the genetic basis of a classic life history dimorphism, the move to planktotrophy to leucithotrophy. And specifically, we can ask, in nature, does adaptive evolution proceed in small steps through subtle shifts in allelic frequency across many loci of small effect? Or alternatively, these are non-mutually exclusive, keep in mind, does it proceed in large leaps with loci of large effect? So in order to interrogate this, we performed whole genome sequencing on 48 wild females from 4 localities, 2 of which are in New Jersey and 2 in New England. And a PCA analysis reveals extensive genetic structuring. So we obtained a very large population genomic data set of over 38 million SNPs that are genotyped in every individual. As you can see along PC1, the morphs are shown with the planktotroph as the triangle and the leucithotroph in the circle. The worms strongly separate by morphology along PC1. The large-egged worms strongly separate along PC2 by geography, whereas the small-egged worms weakly separate along PC2 by geography. And this is a likely reflection of them being in the water column during their growth, so they exhibit higher levels of gene flow. So let's ask, what are the loci implicated in the separation of morphs along PC1? One way you can do this is you can march along the genome between the small-egged and the large-egged females, and you can calculate differentiation. And areas of high differentiation will possibly point to loci that are important for this life history difference. And the logic behind this is, again, that selection acting on different individuals in different locations can lead to adaptive divergence in trait means and possibly allele frequencies. So if we look across these 38 million SNPs, you can see that the morphs are largely undifferentiated across the genome. We have a very large number of loci that exhibit very little differentiation, and this is reflected in the mean FST between the two morphs. And of the 38 million, there are over 36 million that are under 0.2. However, you'll notice that this distribution contains a tail, and there are actually 5,140 loci across the genome where FST is 1 in comparison between the morphs. These are associated with 285 protein coding genes, which represents 1.4% of the protein coding repertoire. So we wanted to dive in a little further, and one of the most obvious places to look is in Hox genes, because they're responsible for patterning bilaterians in early development. Strablospio benedicti has 11 Hox genes, but when we look across the morphs, there are no obvious Hox candidates to explain the life history dimorphism. So to get a little bit more quantitative, we asked how many regions of the genome are implicated in morphological differences, and what is their distribution across the genome. And we took a very simple approach and naively defined an outlier region as a continuous stretch of the genome containing at least one locus of FST1 per 2 kilobases. What you can see is that the median chromosome contains 36 outlier regions. However, chromosome 11, which is the sex chromosome, X chromosome, contains more. And if you compare this to the number of FST values of 1 per chromosome, you can see that 10 pops out as containing a very high number of FST fully differentiated loci. So this suggests that chromosome 10 and 11 are interesting to look at. 11 just because it contains numerous regions, and 11, and then 10 because these regions harbor a very dense repertoire of fully differentiated SNPs. This is the distribution of the outlier region sizes of the genome. The mean size is around 6,000 base pairs, but you can see that there are a few outliers that are very large in the range of 80 kilobases. And this maps to chromosome 11, which again is the X chromosome. So we have to do further work to investigate what's going on there. Just to give you a taste for what genes might possibly be under selection, we found 26 genes with fixed missense mutations between the morphs. I won't get into specifics, but please note that a disproportionate number, again, are on chromosome 10 and chromosome 11. Our colleagues have also generated an RNA-seq data set between the eggs of the different morphs, and we found a few overlaps between their differentially expressed loci and my outlier loci. We have some DNA binding genes, which are zinc fingers, and then genes of diverse function. But while genes are important, we are interested in understanding how these phenotypic differences arise. So what does a region under selection look like, and how can we infer what's happening? So here I have a section of cytochrome P450 26A1. Don't worry about the name, it's involved in retinoic acid metabolism. And the black diamonds represent FST values, while the larger dots in green and orange represent pi, average pairwise nucleotide diversity for planktotrophs and lecithotrophs. And what's important to take away here is that differences in morph-based pi under regions of elevated FST indicate strong selection in the morph that has the locally reduced pi. This is a classic signature of selection. So you can see also in this region, highlighted with the red diamond, that there is a missense variant fixed within the lithotroph population. So the signature of locally reduced pi in the lithotroph coupled with a missense variant makes this a very good candidate for further follow-up. This assumption only works, however, when levels of pi are consistent between the different morphs. And as you can see from this plot, there's no significant differences between genome-wide pi between planktotrophs and lithotrophs. Now we can look to see if this pattern of regions of the genome actually indicate that selective sweeps are occurring in lithotrophs. So one way you could test for that is you could plot FST along the x-axis and then pi for the planktotrophs and the lithotrophs along the y. And if you always see that in regions of high differentiation, the lithotroph has reduced levels of pi, then you can likely infer that sweeps are happening in that population. And as you can see, as the FST values increase, so too does the delta between the between the morph-specific measurements of pi. And you can confirm this with a positive by doing a linear regression. And as you can see, about 20% of the variance in the difference between the morph-based pi is explained by FST. Lastly, the folded site frequency spectrum reveals an enrichment of rare alleles in lithotrophs in the outlier regions, and this is consistent with recurrent selection on the lithotrophic population. So what are the key takeaways? Most loci exhibit small amounts of differentiation in S. benedicti morphs, but thousands of loci across hundreds of genes are implicated in larval morphological differences, and recurrent sweeps are likely operating within populations of lithotrophs at loci related to the larval syndrome. I want to draw your attention to this paper, is that I don't think my finding is that idiosyncratic, that polygenic adaptation with recurrent sweeps is just found in my organism, because in this Littorina paper, they too found a highly polygenic architecture for differences between live-bearing and egg-laying snails. And with that, I would like to acknowledge my lab and NYU GenCore, and I'll take any questions. In the representation of, like, if you go to a field site and you see, so some field sites are exclusively planktotroph or lithotroph, but we also have a number of sites that have mixtures of the two, but we don't see any intermediates, so there's likely strong selection against intermediates, which is maintaining the dimorphism in the wild. Okay, thank you.
Hello, everybody. My name is Shayna Earl. I'm a PhD candidate at the University of Louisville in Kentucky, and today I'm going to be talking to all of you about a portion of my dissertation work that focuses on natural pond temperature variation in ponds around Louisville and how that impacts larval growth and development in the spotted salamander. So as I'm sure we're all aware, our planet is undergoing rapid climate change that's really impacting everything around us, and this includes heat waves. We know that heat waves are increasing in frequency, in length, and intensity. However, most temperature studies related to climate change focus more on increased mean temperatures. From those studies, we know that there are commonly advances in phenology, shifts in the range, populations moving upward in both elevation and or latitude, and also decreases in survival and increases in local and larger scale extinctions. However, we don't know as much about temperature variation and how that impacts many organisms. Oh, I did something, okay. We also know that amphibians are majorly impacted by climate change. As ectotherms, they're incredibly sensitive to the temperature in their environment, and they need that to regulate their bodies and everything going on with them. They have documented shifts in their range and their phenology due to climate change, and also documented shrinking body sizes, and reductions in populations. They're widespread across the globe, and so they are being impacted by climate change and heat waves wherever they are. Metamorphosis is an important life stage step in all amphibians, and their metamorphosis timing is known to be impacted by environmental conditions, for example, density or desiccation. Importantly, it's regulated by corticosterone, or CORT, which is known as a stress hormone, but it does a bit more, and I'll get into that in a moment. My study system are the spotted salamanders. They have a large range across the United States and Canada. They're well studied across this range. We know that the adults migrate to vernal ponds in the late winter, early spring every year, and they deposit egg masses. Those egg masses then hatch, and the juveniles metamorphose in one season. So in this one season, they're in these critical development periods, but they're subject to the environment of the pond. They can't leave the pond. What's happening in the pond can impact them. And so I wanted to know how spotted salamanders would respond to natural temperature variation and what the consequences were for growth and CORT expression. And I wanted to look at this at a single time point early in development when they're especially susceptible to environmental variation. And then I also wanted to track some of these populations across time and see how these consequences persisted across ontogeny. I predicted that with increased temperature variation, you would see a decrease in growth or size of the salamanders. With increased variation, you're more likely to have temperatures outside of their physiological range, and there might be impacts as they're attempting to thermoregulate. I would also predict, for similar reasons, to see higher expressions of CORT. A less stable environment might be more stressful. And I would predict that these patterns would hold across time, knowing that growth rate does naturally decrease over time, but we might be able to see some of these consequences from early growth persist. And we also know that CORT expression naturally fluctuates across development. So as you can see here, it will naturally increase leading up to metamorphosis, and it will peak immediately before metamorphosis and then begin to decrease again. But this can be adjusted depending on environmental conditions. An artificially inflated CORT is known to be associated with differences in metamorphosis timing. So to do this, I tracked multiple ponds at 2 different sites in May of 2023, approximately 2 to 4 weeks after most of these salamander larvae would have hatched, to investigate how this temperature variation was impacting them at this early critical stage in their development. I then followed 3 of these ponds across the development period monthly to see how how temperature variation impacted the salamanders across the development stage. I began tracking hourly temperatures in these ponds in March of 2023 when we found that there were egg masses in the ponds. Salamander larvae typically like to spend their time at the bottom of the water column in leaf litter, so we placed temperature loggers at the bottom of the ponds to be able to get the most biologically relevant temperature data. Once we collected that data, we found the coefficient of variation of the daily minimum temperatures, because daily minimum temperatures is known, again, to be biologically relevant for the salamanders. For this early snapshot in time, we took the coefficient of variation from when we put the temperature loggers in March all the way through to our sampling date in May to have this wider understanding of temperature variation while they were developing in the eggs and then after they had hatched. When we were looking across time, we looked at smaller, more immediate temperature variation for the 4 weeks preceding each sampling date. We also collected zooplankton and macroinvertebrates, as many of those are prey for the salamanders, and it's known that prey availability can impact salamander size, so we wanted to be able to account for that. We identified them to their family level and quantified the known preferred prey. To capture the salamanders themselves, we used dip nets using 1 meter sweeps, and then we calculated the catch per unit effort of the salamanders, which was the overall number of salamanders we captured in the pond, and the number of net sweeps it took to capture those salamanders. After we captured the salamanders, we took pictures of them and used those photos later to assess their length using image J. And we also used those photos to assess their development timing. We used 4 broad development categories: early development, before larvae had any hind limbs present; Middle, when there were hind limbs present, but they're out of proportion with the rest of the body. Late development, where they still have all 4 of their limbs present, but they're more in proportion. They've grown into them a bit more. And then metamorphosis, where the gills have started to lose their feathering and maybe even started reducing in length. At the field sites, after we captured the salamanders, we placed them in water and let them sit in that water for 60 minutes as they naturally express their court. We then put the salamanders back unharmed, and were able to take the water back to the lab and extract and quantified court using established methods that have been validated in spotted salamanders. All right, so let's get into results. How does natural wetland temperature variation impact the growth and court expression in the salamanders? Before we get too far into this though, I want to show you what we had with temperature. So we have on the x-axis here the time, and then on the y-axis temperature. And with one pond, you can see here that although we have an overall trend of increasing temperature as we get into the middle, late summer, there are overall temperature fluctuations. And when we look at another pond that we tracked, you can see that there are also temperature fluctuations, but they're more extreme. So we had ponds that had higher overall temperature variation and ponds that had lower overall temperature variation. When you put all the ponds together, you can see that we had 4 that had this high overall temperature variation and 3 that had the low overall temperature variation. Okay, so now I'm going to talk to you about some of my results from this early snapshot. If you remember, we would predict that with increased temperature variation, we would predict to have smaller salamanders. And we actually found the opposite, where increased temperature variation, in this case about twice as much temperature variation for the most variable ponds and the least variable pond, we had larger salamanders. And we think, though this hasn't been validated yet, we might dig into this, that higher temperature variation would lead to earlier hatching, so the salamanders would have had more time and opportunity to forage before we went in and measured them in May. We also would predict with more prey available for salamanders that we would have larger salamanders, and this is what we found, as you would predict. We did not find that court expression was explained by temperature variation, but I do want to talk to you about development stage. So as I mentioned earlier, you would expect court to naturally increase across development before you get to metamorphosis. And though it wasn't significant, we did see a trending increase of court the more developed a salamander was. So that was exciting to see, even if it wasn't significant. Diving into time, looking at these salamanders across development, we did not find that size was explained by temperature variation, and this is potentially due to the natural decrease in growth rate as the salamanders are more developed after this early development stage. With court concentration, you would expect that it would naturally increase with temperature variation. That's what we predicted. And we did actually find that. Higher variation in the ponds predicted higher court concentrations. But we only had 2 ponds here, and we only looked at them twice. But you can see that this trend does still hold when you look at these 2 ponds separately. However, when you consider month, you see that the July sampling was higher, and so at this point, we're not able to tease apart temperature variation, which was higher in July for both ponds, and also July is when the salamanders are going to be more developed. So at this point, we're not able to parse out this full relationship, but it's something I'm interested in looking in further. And lastly, looking at court and development stage again, we would predict an increase up to metamorphosis and then a decrease once salamanders have started metamorphosing. And like with our early snapshot, we don't see any significance here, but we do see the overall expected trend that we would have anticipated. So in conclusion, in early development, natural temperature variation does predict larval salamander size. And again, this may be because there were higher temperatures, led to earlier hatching, potentially allowing for this longer foraging period. And across time, as I mentioned, we don't see such impacts, and this is potentially due, again, to the natural slowing in growth in these later larval periods. We do see the expected trends in court across development stages, even though it wasn't significant. And it is possible that temperature variation may minimally affect court compared to changes with development, though it's difficult to tease apart in this field study. And so I will be following up with an outdoor mesocosm experiment where I will be manipulating both temperature means and variation to be able to pinpoint the effects of temperature on growth, development, court, and survival, which will help to provide critical insights in the capacity of these salamanders to respond to changing climates and increasing heat waves. With that, I would like to thank the many people who helped make this research possible, my funding sources, and I will take any questions. Yeah.
So I'm wondering, I know that you quantified the variation across these ponds. I'm wondering if you did calculate the mean temperature across time, and whether or not it's relevant for amphibians to have some sort of metric of like total experience temperature. So you're probably familiar, like insects, we use something like growing degree days, and I wonder whether or not that has any relationship to the variation that you're seeing.
Yeah, so I did look a little bit at mean temperature, but I decided to focus on temperature variation here, especially because there are expected changes in mean temperature across time and ontogeny. July is typically a lot warmer than May in Kentucky, but I'll be able to dig into that a bit more with the mesocosm experiment. Yeah.
I noticed that in your graph of the temperature variation, some of the ponds were observed for less time. And I know this is a field experiment. Can you explain why you only had a couple of months for that one versus the entire summer?
They dried up. So, you know, with field experiments, you don't always get what you're looking for. And a lot of the smaller ponds that did actually have higher temperature variation dried up pretty quickly. So I was not able to follow the salamander larvae across the whole summer. Thank you guys so much.
Okay, so before I get started, I do want to acknowledge that this work was undertaken on and using specimens or data collected from the lands of many indigenous peoples across multiple continents. The bulk of this work took place on the lands of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit, and I now have the privilege to work on the unceded lands of the Anishinaabe-Algonquin Nation. Specimens for which I have locality data were collected on the lands of the Chumash, the Tongva, sorry, I forgot I should use the mic, the Tongva, and the many peoples who identify with the indigenous movement in Kenya. Data come from the lands of many, many, many indigenous peoples, too many to name here. And I just want to invite everybody in this talk today to reflect on where you live, work, conduct fieldwork, and where your data comes from. There is a really great tool called nativeland.ca that documents the territories of indigenous peoples, predominantly in North and South America, but they do also have data for other continents. So I recommend taking a look at it and seeing whose lands you are working on and where your specimens come from. So I think we can all agree that life history is a fundamental aspect of any organism's life. And life history is also correlated with other ecological traits as well as things like extinction risk. Now, it's pretty easy to study life history in extant animals because we can actually directly observe their pace of life. We could theoretically tag an individual and follow it from pretty close to birth to its death. However, and unfortunately, most of Earth's biodiversity is dead, and that means that we're missing out on most of the evolution of life history strategies if we restrict ourselves to only looking at extant organisms. So what this means is that if we really want to dig into how life histories have evolved over time, we need to get creative and devise different ways to study life history from the fossil record. Now, as I'm sure you can all sort of guess, the fossil record doesn't make this particularly easy for us. Typically, we only have things preserved as hard tissues, skeletons are almost always incomplete, and we must infer things like ontogenetic stage from, if we have them, teeth. In mammals, we have epiphyses, sutures, or if we're extremely unlucky, relative size. So the question then becomes, if we want to start to look at life history evolution in the fossil record, how do we do that? And one of the techniques that I am particularly interested in is called osteohistology or bone histology. It's been frequently used by extant herpetologists as well as increasingly by paleontologists over the last couple decades. And essentially what this is if you picture everybody's favorite example of growth rings, a tree, you actually have a very similar phenomenon happening in the bones of vertebrate animals. So the bones of vertebrate animals, and in particular the long bones are very good for this, deposit these cyclical growth marks in the cortex of the bone, and these are deposited on an approximately annual cycle. And you put those two together, you're like, all right, 1 ring per year, cool, just like tree rings. What's more, we have developed ways to estimate body mass using the circumferences of bones. So if we have these two things, age and body mass throughout life, we can get a growth curve. So we can start to look at the actual adding of mass, basically, of these animals throughout their lifetime. And there are many things that we can do with those growth curves. We can look at things like age at maturity, which is a very traditional life history trait. We can start to look at how body size has evolved within different clades. And we can also look at different things like physiology and metabolic rate. But one thing you may have noticed is that all of those last slides pretty much used examples from dinosaurs and birds. So the most rigorous methods for doing these sorts of reconstructions of growth curves have never really been tested properly in fossil mammals. And we might expect mammals to be different. And especially, it was actually thought for a very long time that endothermic animals like mammals would not deposit these annual growth marks because they're not as dependent on environmental conditions for maintaining homeostasis. However, there was a really great paper in 2012 by Kohler et al. That showed that a global sample of wild ruminants have arrested bone growth during the unfavorable season. And this occurs in tandem with decreased metabolic activity as well as decreased levels of the growth hormone IGF-1. So we know that growth curves are deposited in mammals and they're similarly reliable to the ones in herps. However, the current state of study of fossil mammal growth curves is still lagging behind the state in other taxonomic groups of vertebrates. So one of the major concerns is that bone is a living tissue and it remodels over time. And this has not necessarily been accounted for in previous studies. There is also little to no curve fitting or model testing of which growth models actually fit the data best. And only a few mammalian groups have been studied, mostly large ungulates. So what I am really interested in is the carnivorous mammals. And we did a very quick sort of, do they even preserve good growth marks? We found that yes, they do. So that was great. This sort of set the pace for my entire PhD, which was awesome. It's nice when your proposed PhD seems like it will work. So the objectives of this study were to validate growth curve reconstruction in extant felids, reconstruct body mass growth in a fossil cat, so Smilodon fatalis, which is a saber-toothed cat, using bone histology, and then test a hypothesis that has been outstanding about Smilodon, in this case, whether it was sexually size dimorphic. So what I used to do this was I first collected observational, so age and weight data for 71 different species of wild and captive feliforms. In this talk, I'm going to be focusing on Panthera leo and Panthera tigris because they are two cats who are pretty much the closest we can get in size to Smilodon fatalis looking at extant felids. I also thin sectioned 1 femur each from Panthera leo and Panthera tigris, as well as 19 femora from Smilodon fatalis, all of which came from Pit 3 at the Rancho La Brea tar pits in Los Angeles, California. And those Smilodon femora represented various ontogenetic stages as determined by epiphyseal fusion. So we had both juveniles and adults. What you do with this curve fitting is, let's say we just have the observational data. This is pretty easy. What I did was I fit several growth models to In this case, it was just the observational data, but you could also do this to the circumferences of preserved growth marks. The models I fit were using the Richards family of models, which are a set of named models that only vary in their shape parameter, and then selected the best fit models using AIC. For histological specimens, things get a little bit complicated, and you do have to do some additional steps in order to estimate the number of missing growth marks. So this is done by essentially projecting the model back to neonatal size and using that to estimate how many years are missing in your sample, and then adding that estimate to the growth mark count and calculating the age at death as well as the final growth curve. So we'll start off by looking at the observational data for the extant lions and tigers. So with tigers, you've got captive animals on the left and wild individuals on the right. As you can see, there's a lot more data for captive animals than there is for wild animals. That's to be expected. So one of the first steps here was checking, can we use these data from captive animals to actually get the information we want? And what's amazing is that it turns out that we can. While there are slight differences between the two curves, there are no significant differences between captive and wild curves, suggesting that we can use data from captive individuals to pretty accurately represent what's happening in the wild. Looking at lions, we see the same pattern. Slight differences in the curves, but there's no significant differences between captive and wild curves. One thing that we recover with both tigers and lions is things that we would expect, like there is sexual size dimorphism, and you can see the SDR listed up on the top, so males are larger than females. We also find that lions grow slower than tigers do. The next step is to do a growth curve based on histology, in this case of a lion, and check to see if that fits with what we actually observe in our data. Lucky me, it does. So this was actually, I got this graph and I was like, yes, because it just like sits very nicely in the middle of the cloud of data points for extant lion observational growth. And as you can guess by looking at it, there is no significant differences between these two curves. Then we have to get into the fun, meaty stuff, looking at reconstructing growth in the fossil species. So out of those 19 specimens I had, 4 of them had data that was useful for growth curve reconstruction, which meant that I needed 3 or more growth marks, and the data needed to capture the inflection point and the slowdown of growth. I'm happy to talk with anybody about why it doesn't work if you don't, but I don't have time to get into it right now. When we plot those 4 individuals, this is what it looks like. The best fit model here is a Gompertz model for all 4 of them. And we can start to look at growth parameters. So the inflection point, so the point at which growth starts slowing down, is at approximately 1 year of age. The animal reaches a body mass of about 235 kilograms. It reaches 95% of asymptotic body mass at somewhere between 2.25 to 4.5 years, and has a maximum daily growth rate in kilograms per day of about 500 grams per day. So 0.5 kilograms per day. What we then did was try to see if their variation in adult body mass, so that asymptotic body mass, correlates with dimorphism in extant. In this case, extant Panthera and Hyenidae. We did find a positive relationship. So basically the larger the size standardized variation in asymptotic body mass, the greater the degree of sexual size dimorphism. And this suggests that knowing the size standardized variation in body mass can help you predict whether a fossil species was sexually sized dimorphic. So, of course, we took our data from Smilodon and plotted it on here. This shows that Smilodon fatalis was moderately sexually sized dimorphic. It likely was more dimorphic than any species of hyaenid and was within the range of dimorphism seen in extant Panthera, but on the low end. So the sexual size dimorphism in Smilodon fatalis is only greater than the snow leopard, which is the least dimorphic of the extant pantherine cats. So the takeaways thus far are that captive animals can be useful proxies for growth in the wild, which is amazing in terms of getting lots of data. Osteohistological growth curves accurately reflect growth patterns in an extant population, also excellent. And what we found was that Smilodon fatalis was slow growing relative to extant cats, and it was also sexually size dimorphic. So, as I'm sure everybody who has ever done any amount of science can relate, I had a lot of things that I want to do now that I've got these data. So one of the things that I am very interested in is looking deeper into the role life history may have played in the extinction of Smilodon, as well as other Pleistocene carnivores. I would like to explore how growth patterns changed across the Pleistocene-Holocene boundary in the species that survived, as well as how it might have changed in response to human encroachment in the historic record. I would like to test for correlations between these types of growth parameters and traditional life history traits like age at sexual maturity, age at independence, weaning, things like that. And one thing that I would love input from the audience, if anybody knows, has any experience with this, is doing ancestral state reconstruction of some of these growth parameters. It's not something I've ever done, but I think it could be really interesting, and I would love to talk to somebody who has experience there to find out more. So thank you all very much for listening. Thank you to all these people, and if you would like to get in contact with me, there's contact information up there. I think I have time for one question while we transition. I didn't see who was first. Who's better?
I was curious, maybe we already talked about this, would you know which direction the sexual size dimorphism is? If hyenas have females larger and the felines usually have males larger?
Not from the type of analysis that I did. There was one outlier that was like outside of the confidence interval on that, and that was the spotted hyena, which is the one where females are larger. So I suspect that there might be different relationships with female bias versus male bias, and that's something I would like to test, but with the data that I presented, it's just they were all male biased except for the spotted hyena, so I couldn't test them. It's a great question. I would love to test it further.
Okay, welcome everyone. I'm Milan Vrtílek. I am a researcher at the Czech Academy of Sciences, and today I would like to talk to you about our project on parental senescence effects. First, some terms. Age is just a number, and by senescence, I mean the decline in physiology and reproduction that is related to age, and it naturally varies between individuals of the same population because it has partially genetic basis and partially it's influenced by environment. How did senescence evolve? There is this concept of selection shadow, meaning once individuals start to reproduce, the selection on their phenotype is declining, and therefore any further maintenance of the body is not that important. An interesting aspect of this is that as there is a variation between individuals, there is also variation within individuals, between different traits. As this theoretical work by Morat and Newsey proposes for these traits related to reproduction, wherever there is senescence in or age-related decline in fecundity, the selection on maternal effects will tend to decline faster. Why to study the parental senescence effects? It's important as the potential generational transmission. We have a few channels how this can happen. One is the random genetic mutations in the germline. Bit complicated is the effect of parental care that can partially buffer for this negative effect of these random mutations. So despite there is senescence or mutation happening at the genetic level, parents can still buffer for that by improving their parental care as they get more experienced. But to me, it's most interesting is the non-genetic factors that should be systematic and to some extent also predictable. We study this on this super interesting fish species, African annual turquoise killifish, that is adapted to this seasonal environment of desiccating pools. So the fish developed this, evolved this condensed life history. So they mature really early, and even in the captive environment in the laboratory, they die naturally after a few months. On the other hand, they survive desiccation of the pool in the state of embryos, and they are able to sustain their life functions for multiple years. Our main question of this project was if there is this number versus quality of offspring, as we expected the fish to improve their fecundity with life, but at the same time we expected to observe decline in the quality of the offspring. We did this in the longitudinal laboratory study where we held this population individually, and we measured traits like the growth and reproductive performance, like egg number, fertilization rates, egg size. Alongside with that, we also collected fin tissue for cellular markers. We decided to sample offspring at 3 specific ages that are related to the survival in females in this population. The first point is soon after the fish or female get mature. That's the age 1. The second one is related to the 50% survival of the female in this population, and the last one is only 25% survival. We collected the offspring in the pre-specified manner, breeding always the same individuals within these family groups, and then incubated the clutches on the dry substrate, like humid, semi-dry substrate of peat for 6 months. Along with the survival of this dry incubation, we also measured their initial growth during the 4 weeks of their life, because at some regions the pools can be really ephemeral, and they sometimes don't last even as long as 4 weeks. So the growth of the juvenile maturing fish is really important measure of the fitness. So now I would like to show you the first results. First part is related to the senescence in the parental fish, and the second part is how did the age of the parents affect their offspring performance. Here you can see the growth. That is sex specific. Males are larger. They reach their final size earlier than females, and female tended to grow until the end of the experiment. Here on the bottom you can also see the period where we collected the clutches for the next generation, and also the X's mark the fig lip sampling. Here is the first trait where we looked at the age-related decline. It's body condition index. It's something like body mass index, which means that lower values on the y-axis suggest that the fish is in a lower condition, and you can see general pattern is both males and females decline. Here in the reproductive parameters, it's more illustrative, and you can see that both in the fecundity and the fertilization rate, there's really a lot of variation, but on average, we can identify moments where the fish get senescent, not so much in the egg size, which can be thought of as an investment into the next generation by females. So for the offspring traits, we saw that the age when the parents produce the offspring negatively affects their survival of this extended incubation period. And we expected something similar for the initial growth of the offspring. This is the expected trend, but neither in females or males we could observe such a pattern. So the size after these 4 weeks seems quite comparable between these 3 groups. We are now working on the processing data for, as I mentioned, we collected also think lips from the parents and from their offspring at the end of their 4-week growth. And we are working on DNA methylation using this DDRAT sequencing method. That is work of my colleagues, and Malahat already presented the data on Saturday. And we accompany this methylation also with the telomere attrition. That is the work of our colleagues from neighboring institute in Brno in Czech Republic. So I have first data on the telomere length, and from that it appears that during the aging the telomeres really get shorter in this fish, but sadly, or I don't know, maybe luckily for the fish, we didn't see this pattern to be transmitted to the offspring. So just to summarize, we identified multiple traits where the parental fish showed age-related decline. Most importantly, they negatively affected offspring survival, but not their early growth. So it can suggest that once the fish survive the dry period, they are basically the same quality. So the survival can be condition dependent. With that, I would like to thank my colleagues for the inspiration and the work we did together, and to you for your attention. And if you have any questions, I'm happy to try to answer. Yes, please.
Do you know if if the senescence of the mother or the father has any more of an impact on the offspring, or is that something that potentially you could look into?
We definitely have the data for this, but I cannot provide you with a definitive answer. But yeah, this is really interesting to really separate the effect of different sexes of the parents. So that's one of our aims. Yes, please.
Cool stuff. Did you detect any family differences? I'm curious
Uh-huh.
if you said you measured 8 different families, and if there were any family effects of the senescent traits, or I guess it would be the offspring traits.
You mean if some trios of the fish were like performing better than other trios? That's definitely possible, and I can tell you that we observed something like this, but I did not quantify that. And it was based on, at the beginning we assigned the individuals with different genetic background to different families, so that was partially kind of random. So I don't know if that is answering your question. Yeah. Okay, if there is no other question, yeah. You can think about it and email me. So.